yeah, also warm welcome from my side to today's webinar. In this webinar for advanced users, I hope that you already used the PlexTyper, or maybe you're just interested in some more functions. Then I want to invite you to different topics today. So I want to talk about how to deal with the basic and the critical warning, and also want to show you how to easily bring a lot of samples into your plate layout by using the Excel work list. And I also want to talk about the quality score. So as my colleague Lily says, just feel free to ask any questions during this session here. You may know that PlexTyper is used to analyze our different products from the AreaQ product line. So for blood group typing, also for the FastQ product line, which is actually for the FastQ CD kit, and also for the FastQ B27 products. And also you can analyze with this PlexTyper more complicated, more specific data from the Rainbow product line for HLA antibody detection. And we are also always want to bring new tests to PlexTyper and also integrate your customer wishes. So if you have one of them, then please tell us, and hopefully we can then integrate these wishes to PlexTyper. So for example, we also want to add another FastQ test, hopefully at the end of the year. We will launch it for the FastQ RHD fetal one, that you can analyze also these fetal FastQ RHD fetal product with PlexTyper. With this, I start here at I have to share my screen firstly. So this one here.
Yes, we can see your screen.
Perfect. Okay. I will start here on this PlexTyper desktop. I think you may know this. And I jump here to this function under admin, these kit releases function. Because I want to say some words about these database here. Here you will find all your uploaded kit files. These kit files are somehow like a data package where the different thresholds are integrated for the different product and also for the different lots. So whenever you got a new lot, then you also have to download the specific kit file from our website. You can also do this here in this kit database with this load kit function here. So you can just maybe go to your downloads folder and then upload a kit file with this kit name here at the end. Then you also may know from the basic webinar that you cannot delete the kit files and also not data from PlexTyper. It's only possible to archive them. So by clicking here on this not archive, then it switches to this green one here, and then it will not be anymore at your plate setup. And you just will see your unarchived kit files. So whenever, I don't know, you do not need this kit file anymore, or the expiry date is over the time, then you can archive them, and then it's not on your test setup anymore. You also have the possibility to filter after these files with this text search function here, or you can filter here, for example, after release date, after the instrument type, or the kit description. With this, I go back to this desktop field here. and go over to view tests with no associated results. Because some few words about this one here. This is also like a database where you can find all your created plate layouts, which are not connected to your raw data at this moment. So with this, you can find all your created layouts. You can reopen them when I just double click here on this layout, for example. And afterwards, you can import your data, your raw data, for example, this Excel file, and then it switches from this view test with no associated results to these view results area here. Again, you can filter after different names, the kit name or the product number, whatever you want. And this is somehow for users which are creating more than one template, for example, print these templates, go to the laboratory, perform the test, and later afterwards they can reopen the different layouts and connect them to their PCR raw data. With this, I go to the view results database. So this, I think you also know, this is the database where you can find all your run tests. So what you can see here is the run ID from PlexTyper. You know that whenever you create a new test, then PlexTyper gives this test this new PT number. So when I will create a new test in a few minutes, then you will see that is PT231. You can again after filter different things, and maybe one thing I want to point out here is these somehow new field, the tech approved and the medical approved function. So what you can do is when I, for example, reopen this run here, you and maybe your colleagues can do the technical improvement by clicking here on this button, or a single for each of these genotypes or reactions, how to say, and then another person can do the medical improvement, and then you will see here in this view results area that this switches then to this green color. You will see that here the technical and the medical improvement is done. Whenever only the medical improvement, for example, is missing here, then you see that there is a different color, or when you have the medical and the technical improvement not done, then it's like this gray one. So that's up to you, this function. You will see also these improvements in the audit trail of your report. So maybe this is an interesting tool for you. With this, I jump over to this tests area here, and want to create a new test here. So you know that the sample ID is needed for PlexTyper, which is then connected to any of my kit files. I create a sample 1000, for example, and connect this to, for example, REQ ABO variant. Then you see that this button here switches to this green color, and then I can place this sample on my plate. But here you can see this warning occurs. And that means that the kit file here, for example, is expired at the end of April this year. And you have two options. You can click no, then you can choose another kit file, or what I will do is clicking yes. If you want to analyze all data, you can also do this with this expired kit file, but you will also see this warning that the kit file is expired on the report. So you can see that this ABO variant REQ test needs 16 reactions or wells, and yeah, there are some other kits, for example, the B27. So I choose the same sample again and just show you quickly for B27. Or I use the B27 direct one, and see that this is only one well. I can go further for other samples and load them on the plate. But what you can see is if you somehow create a lot of samples for B27 direct, for example, which just need one reaction, or the CD test, which need 2 reactions, you need a lot of time to click all these samples on this plate here, always right here in your sample ID. So there are 2 options to do this more easier. One slight thing is that you can add to your computer and barcode scanner, scan the barcode of your sample tube, for example, and then the whole number appear here. This is one little option, but the main thing I want to figure out here is the Excel work list. So with these 2 buttons, you can somehow create with this one here the Excel work list, and with this you can import this Excel work list. So I will do this now. You have the option to save this Excel work list on your local desktop, or in any folder, or you can do this on a secure server, which is secured by a password you have to choose. I will do this with this local server here, so just on my desktop, and use webinar 24/6. Save this. Then I just make this window here smaller, and just look at my desktop for this Excel work list here. So I just double click on this and open this file here. And what you can see is just the basic table. So you have here the sample information and also the kit information. What you can do under sample ID again, which is here in red. So the sample ID and also the kit file again is important for PlexTyper. And what I now can do is creating sample 100 again, and click here to move these samples here down. And what you can see is that I create 20 samples in a few seconds, give them also a specimen type, blood in this case for the B27 direct, and choose the correct B27 kit file. You can do like in text search, and I will use this kit file here. But just be careful when you will do this for all the samples. Do not move this kit file like this, because otherwise you can see that the ID here moves, and this is then not the correct kit file anymore. So I just copy this one, mark this whole field here, and then with a strong integrate, then you can fill in here this whole table. You can reuse this Excel work list for different runs, but one important thing is you have to be careful when you upload a new kit file to your PlexTyper. Then it is important to download again a new work list, because otherwise this kit file is missing here in this table. So then I will save this Excel work list. And another important thing is that you have to close this Excel work list before you can upload this in PlexTyper. So yeah, I move back to my PlexTyper software, and now you click this arrow. I open from my local server the webinar work list. And then you get this remark here that you have to be sure and check the plate of the positioning, and also the samples are on the correct position. I click OK here. And then again, what you may know is you can print this layout as a picture like this, or as an PDF where you have this like table. I can show you shortly, which is, in my opinion, the better option, because you will see the sample name and also the kit file. I just move this here to my window. So yeah, it looks like this, where you get the address of the well position, the kit name, also the lot, and my sample ID. And this you can print and work with this in the laboratory. All right, before I move further, do you have any questions regarding the Excel work list?
I encourage you to ask a lot of questions. Use the session, use the knowledge of my colleague.
Okay, if not, then I go further. So the next step will be that you just click this play button. I think you already know this, and import your raw data file. But I want to go in more detail to different results. I think I will start with this one here. So this one was like an KKD MNS kit file or run. And what we find here, I think you know this window, but I just want to go over this. Here the positive reactions, the negative reaction, and here in green, the internal amplification control. And then you see here these different colored bars. These colors stand for the different fluorophores we used in this assay. So the orange 560, the quasar, and also the red 610. And then you can see that the bars are located after the quality score. You can switch this to the reaction number. So it goes now from 1 to 8, or from A10 to H10. So that you can guide over these different reactions. But today I want to talk about this quality score. So you can see here on the left side in yellow, for example, this bar, which has the highest score for the positive reactions, which is 10, and goes down to 0. And for the negative reactions, from 0 to -10. And that means that you have like the reaction quality, which is based on different parameters I will show you now. We will start here with this, maybe I will just also go over these. When you go with this mouse over these bars, you can see the well position, the quality score, right? And also the specimen of this reaction. So it switches when I move here, for example, here, Kel, and so on. And when I double click on this bar, for example, so we start with this quality score 10, a really good positive reaction. You can see here this data review chart. You will find different colored points here. So we have here in green, somehow in data cloud, and these green points are positive data, positive reactions from different samples which you have run with the same kit file, and which is for the same reaction here. In blue, you can see your reaction. Actually, this one I have double-clicked on. And in red, you can see here and here the negative calls. When I double-click here on this blue dot here, you can see the target fluorescence, the target amplification curve, and here in green in the FAM channel, the internal amplification control. So PlexTyper calculates with the 3 different parameters which are important. So one is the CQ ratio of the target, and the target CQ is divided with the CQ of the internal amplification control. So for example, 27.37 with 24.67. And then you will see here when I go back in the top of this window here, the CQ ratio to control, which is 1.11. And this has to be located between the minima and the maxima target CQ ratio threshold, which is between 0.95 and 1.27. All these reactions here are located in this area. So for example, this one here has a ratio of 1.16 and so on. Then the second parameter is here the final fluorescence. So the positive reactions have to be located over these border here. And then the last parameter, which is important for the positive reactions and also for the quality score itself, is here the shape of your target curve. So PlexTyper looks how this curve here statistically looks like in perfect sigmoidal curve. If the shape of this curve is like this one here, like this sigmoidal one. And these 3 parameters lead to the quality score and can differ. For example, if the fluorescence was too low, or if the CQ was a little bit too low or too high, and lead to the different quality scores in this case. Here it was just a really good positive one, but we also look at the best case for a negative reaction. So you can see when you open this window, again here in this corner, you will find in blue your sample or your reaction. And what you can see here is that there is no amplification for your targets at all. You only can see your FAM channel, so the internal amplification control. So, but now we go more into detail of these tools here. You can see this blue mark here or flag, and also this red one. The blue one means this is the basic warning, and that means no CQ could be calculated. That means, so we go over this one, that you can see that your reaction is located here. So when you check it, you can see, okay, this reaction is far away from the area we look at, positive reactions. So this is like a good sign. When I double click on this, you can see that there is no CQ calculated for your target curve. You see that there is a really late increase, I think after 39 or 40 CQs. And this is like a strong increase here, which we sometimes see, which is in classical artifact, or which maybe is based on primer dimers, so that the reaction is over here. So we also have some of the reactions which we called a road runner, which go over here and move to this area. So for example, this one looks basically like the same one. So PlexTyper calculates them correctly as negative ones. the more critical one, and I also want to point out that you have to look at these critical warnings where it says close to threshold. And what you can see here when we double click on this one, that this reaction is located quite here in this corner, directly on these two different borders. When I double click on this reaction, you can see, okay, here is my internal amplification control, which is almost there with an CQ of 24.98. And then there's a slight increase here in blue for your target, but you have this CQ here. I mean, okay, the curve looks not really nice, and there is also just a slight increase of your target. So this can be because of different things, because the direction is not optimal, the DNA concentration was too low, or the quality of this sample was not that good. It can be that the fluorescence was too low, for example, pipetting issues, or just that it is for this one, for example, it could be that maybe because of the test design, different probes are a little bit lower compared to others. And then when you have a little bit less quality of your DNA, then it can look like this one here. So please check these reactions. Probably if you have no results here in this summary, then you have to repeat this, check again the concentration and the quality of this reaction. But what you can do, for example, in this case also is when you go here and click show alleles with positive calls. So PlexTyper says for this reaction here with this specimen, this one was positive. I mean, with a bad score, right? But PlexTyper says, okay, this is a positive one. What you can do is when you click on the right mouse and go to clear alleles, I do it again, show alleles with positive calls, then you can see here somehow a list which reactions have to be positive for these genotypes here. And when you see this, you can compare your reactions. So I look at this one here. You have here, or you have to be here a positive call, and also this one have to be positive. So when I look at this reaction again, you can see this is like a really good positive example. And for this genotype, PlexTyper needs this as a positive call and just also this one here. The next different reactions here have to be negative for this. And that's why, which is somehow sometimes a good indication that you can compare your results with these show alleles function to find out, is it a correct positive result or not? Do you have to repeat your samples? So my conclusion to this warnings here, especially this critical warning, these are somehow marks that helps or that have to show, okay, I have to look manually on this curve here. Then maybe one other example is this one here. Maybe you already had this example where you can see that the internal control is missing at these two wells, and also you got no results for your targets. You also can see this here in the genotype that there is no results and you got no predicted phenotype. And that's why here in this case, probably the sample was not pipetted in this well here. Maybe it was go in this further on in this well after this one here. And that is an indication that you have to repeat your test because also you got this remark here that the internal control failed. And then another one for B27, because we already talked about FastQ B27 Direct. You have the option here for this area to also look after only the common alleles, the common and well-documented alleles, which are the well-documented ones in blue, and also all alleles, which are also these rare ones here in gray. So it's up to you what you want to report. I think most of the customers do this common and well-documented ones, especially also for rainbow. And what you also have here for B27, CD, and also rainbow is these mismatch area, where you can annotate your results. If you see that you got no results for one target, for example, that you have to recheck one of the reactions, which is maybe too low or which has a warning there, then you can go and switch these mismatches from 0 to 1, for example, to look if you can annotate one of the reactions or not. If the problem is, for example, the sample or not, if you have to repeat this test, so this is also like a good tool here in PlexTyper to annotate your mismatches. I jump back to this sample here because I want to show you how to create different types of reports. What you can do is when you create your report, you can set report file name formats. And with this tool, you can change the name of the report. So in my case, I just have the sample ID here, and then the report is saved after the sample ID. Or you can add, for example, I don't know, the timestamp, and then the name of the report changes from only sample ID to sample ID slash or this stripe here, and then the timestamp. You can then generate a report for all of your tests or just for this reaction here. So for this sample. And what you also can do is with this tick here, if you want to have this audit trail in your report or not, then you can click this here. If you don't want to like this, I mean, for me, it's interesting, or most of the customers also use this because you can see which or how what the people did. Do they look with the technical and medical validation, which people or if the people create the layout or not. So this is also interesting. Then you can export the data with different options. So you can also export the data as a text file, as an NHSBT file, or as an HTML file if you want to. Especially for the HLA users, maybe this is interesting. And maybe one last thing here is you can also print again this plate layout or send this layout if you have any problems with your runs and we need these information of the plate layout. Then you can export these files as a PDF table again and save this on your computer. All right. With this, I think I'm done for today. Do you have questions, or maybe I forgot something to explain? Please ask.
Let's see. I don't see any questions in the chat, but you can still simply raise your hand and I will activate your microphone and you can speak and ask questions. I would like to have some interesting questions. Well, that's unusual. I don't see any. Meanwhile, I will post our contact on LinkedIn. So if you are on LinkedIn, we would be happy if you connect with us. And the second one, if you don't have any questions, then we have some for you. And I will post the link to these to this quiz, so you can request your certificate of attendance. 2 simple questions, and then you will receive it automatically. Well, I don't see any questions. That's really unusual. But that means we can say bye, and we would like to see you in our next webinars. We haven't announced them yet, but we will have more webinars in the second half of 2026. I wish you a nice day. If you have any questions afterwards, you can always write us an email, and you are going to find the contacts on our website or in your email client. Have a nice day. Not too hot, because it's very hot in Germany. Bye-bye.
Have a nice day. Bye. Thank you.